Good afternoon, and welcome to this afternoon's presentation on Medihoney. Honey has been used in wound healing for centuries. Evidence for its wide range of medicinal use has been found in ancient writings such as Edwin Smith Papyrus in the 17th century BC. However, only in the last decade have microbiologists begun to understand the uniqueness of the Leptospermum species. Today we have Professor Rose Cooper, who will provide an in-depth review of her research in Manuka honey and the impact it has on wound healing. Lisa Sangster will follow Professor Cooper with how she leverages the unique scientific properties of Medihoney to optimize the healing of her patients' challenging wounds. Professor Rose Cooper recently retired from her role as Professor of Microbiology in the Department of Biomedical Sciences at the Cardiff School of Health Sciences. Professor Cooper began to investigate the microbiology of wounds in 1992, which led to an interest in the efficacy of topical antimicrobial agents. Since 1997, she was especially interested in studying the mode of action of Manuka honey on antibiotic-resistant strains of bacteria isolated from infected and chronic wounds such as MRSA. Professor Cooper also spent her career investigating the potential of bacteria recovered from wounds to form biofilms and searching for ways to limit biofilm formation. More recently, she tested combinations of antibiotics and honey on wound pathogens to find synergistic interactions. In collaboration with Professor Chris Griffith, the role of hospital environmental surfaces in the transmission of bacteria has been explored, with a view to limiting cross-infection by effective cleaning. Lisa Sangster is a clinical nurse educator for the Nova Scotia Health Authority, covering 5 rural hospitals in northern Nova Scotia. Lisa's clinical practice spans the fields of emergency nursing, cardiovascular surgery, pediatrics, and long-term care. She's a self-taught wound care clinician who enjoys assisting in the care of complex wounds. I believe you'll find their presentations interesting, informative, and quite compelling. Professor Cooper.
Thank you. Well, I'm going to divide my talk into 3 parts. I'm going first of all to talk about the characteristics of honey, then I'm going to talk about its antibacterial properties, and I'll finish by looking at its possible impact on wound healing. First of all, I'd like to thank Integra for the opportunity to come to Toronto to give this presentation. As Andrew's already said, honey, of course, is an ancient remedy for wounds. It's been used for thousands of years. Certainly it was used in British hospitals up until about the 1970s, and nurses would bring jars of honey onto the ward. At that time, antibiotics were very effective, and so ancient remedies began to wane, really, interest in them. There's the development of more sophisticated wound dressings began around that time too. So honey kind of got set aside. The first modern licensed wound care product containing honey was actually developed in Brisbane, and that was licensed in 1999. And since then, during the 21st century, we've seen tremendous advances. New products being developed, some of them from ancient remedies, some from new, and Health Canada approved Medihoney in 2007, as indeed did the FDA. So we've come a long way in this century. We now have what I call second generation products. We're not just using sticky brown honey. It's kind of formulated into advanced dressings, which are relatively easy to use and not necessarily sticky or unpleasant in any way. Now, we wouldn't use table honeys today, and there are very good reasons for that. I did a survey for Dermasciences a few years ago, where we were asked to look at the characteristics of some table honeys. So we went to supermarkets and bought some table honeys. We tested them in the laboratory for their antibacterial activity. It's a test that had been developed in New Zealand. And we had one reference honey, which was a medical grade Manuka honey. That was our reference point, and that was the most active agent. Of the honeys that came from the supermarkets, 10 of them had absolutely no activity that was demonstrable in the laboratory test. And 3 of the honeys were Manuka honeys, are labelled as Manuka honey, and they didn't even actually perform as one would expect from the label. And we now know that there are in fact honeys that purport to be Manuka honey and are not Manuka honey on sale for consumption. But the biggest risk of table honey is that it contains bacteria. And when you plate it out onto agar, you find a whole range of spores, predominantly soil originating, but you also can get things like clostridial spores. And something like about 8 to 10% of honeys will contain Clostridium botulinum, which of course, if introduced into a wound, have the potential to cause wound botulism, which of course one would not want in a healthcare establishment. So there are a wide range of honeys that are used in treating wounds these days, and the kind of honey really depends on the country. The picture that you see on that slide is in fact a Manuka plant, and Manuka grows extensively in New Zealand. There are also about 80 species in Australia, and they are the same genus but not necessarily the same species. So Manuka from New Zealand, commonly known as the tea tree plant, not Melaleuca, which comes from Australia, is a Leptospermum scoparium. And there you see a honeybee foraging on the Manuka flowers. What we do have these days is what's called medical grade honey, which may be suitable for clinical use. Now, if you're going to find a medical grade honey, you need to know that it actually has proven antibacterial activities. And as I've already said, there are laboratory tests where you can look for zones of inhibition in the laboratory, and you compare the activity to phenol. Now, phenol is a reference antimicrobial agent that's long been used in evaluating antiseptics, so it has a long provenance. Medical grade honey should be collected under hygienic conditions, and so the beekeepers need to be trained to collect it under clean conditions and to minimize its contamination. It must be, if it's going to be incorporated into a medical device, it's got to be traceable. So that means it has to be labelled at the source of production, and that must be traceable throughout the whole of the production process. Advisably, we want the honey to be clean, and what I mean by that is that it should come from unpolluted areas, must come from healthy hives, not hives where the bees have been had a disease and been treated with antibiotics. And certainly we want it to come from remote areas where we're going to get least chemical residues or pollutants in it. But the most important thing about medical grade honey is that it is sterilized by gamma irradiation. So you see that plate in the corner of my slide there. We've spread medical grade honey onto that plate, but nothing will grow. It doesn't mean to say you can take any honey, sterilize it, and it's going to be a medical grade honey. So it's got to have the various attributes that I mentioned on that slide. Now, honey is a natural product, and it's a very complicated product when it comes to its chemistry. Some factors are common in all honeys, but then there are some that are unique. Now, if we look at the various antimicrobial, antibacterial factors in honey, all honeys are roughly about 80% sugar by weight. Now, that will be antibacterial, but certainly when you put it onto a wound, the sugars rapidly get diluted and lose their antibacterial components or potential. Neat honey has very low water content, and in fact that will never allow the growth of bacteria. So honey will be able to be kept for long periods of time. It also has low acidity, and that too is an antibacterial factor. Now, in the 1960s, some work was done in Cornell University, and Jonathan White's group discovered that there was an additional antimicrobial factor in honeys. They looked at 490 honeys and found that they produce hydrogen peroxide. There's an enzyme in the honey that oxidizes glucose and produces hydrogen peroxide. So that was a mystery component that it took a long time to discover. Additionally, in some work that was done in the Netherlands, an antibacterial factor was an antimicrobial peptide, and this is bee defensin. This is part of the immune system of a bee, so it protects the bee from being infected. Also, there are lots of antioxidants, polyphenols, flavonoids and terpenes that come from the plant where the nectar's been collected, and many of those also have antibacterial properties. So of course bees go to lots of different plants, and so there will be lots of those components in the honey. But one important factor which is unique to Manuka honey, and most of my talk will concern Manuka honey, is methyl-glyoxal. Now this was discovered in a study that was conducted in Germany, and it is a heat-stable compound, so it's not destroyed by heating a honey, as indeed the enzyme that generates hydrogen peroxide is, and it's become known as the unique Manuka factor. So from now on I'm going to abbreviate methyl-glyoxal because it's a bit of a mouthful, and I'm going to call it MGO. So the unique factor, or UMF as it was called in New Zealand, comes basically from the MGO. So as I already said, it was discovered in Germany and confirmed by a study that was conducted in New Zealand. So the UMF correlates to the MGO, and two studies confirm that. One conducted by Julia Atrott and Henley, and another one by Constantin, which was actually on 80 leptospermum honeys from Australia. What kind of UMF should you use for wound care? Well, in a review that was written a few years ago by Molan and Tanya Rhodes, it was stated that the FDA have recommended a UMF between 12 and 16. And certainly Medihoney has a UMF that's greater than 12, so it's 12 plus. And that will correlate to 355 micrograms of MGO for every kilo of honey. So this is it is an unusual and unique factor that's found in leptospermum honeys. Other honeys have very, very low levels of MGO, if any at all. Now, let's turn our attention now to the therapeutic claims for honey. And this is a review that was published in 1999 in the Journal of Wound Care by a great friend of, and mentor of mine, now the late Peter Molan. And what he did is he drew together evidence of the antibacterial and the wound healing characteristics of honey. He did this by looking at all sorts of papers, and a lot of the those claims at that time were not strongly supported by objective data. A lot of the evidence at that time was anecdotal. But I'm hoping that by the end of this evening you'll see that that is no longer the case. So essentially I'll look first of all at the antimicrobial activity, which is, as a microbiologist, where I first came into the story, and then I'll finish off looking at the wound healing characteristics of honey. So when I first started doing this work, it was with a couple of honeys that Peter provided me with, New Zealand honeys, and I had started to build up a collection of clinical isolates, and so it seemed logical to see if they were susceptible to these honeys. I have to say that a lot of work on the antibacterial properties of honey had been done before I got involved, and a review was written in 1992, but the trouble was there were so many different honeys and so many different techniques, some described better than others, and a lot of the work had been done with reference organisms, that really it was a very confused picture. So I stuck more or less to Manuka honey, and I stuck to clinical isolates. And I chose two main groups of bacteria. First of all, Staph aureus, because it's the most common cause of wound infections, and secondly, Pseudomonas aeruginosa, because that too is a difficult organism to treat clinically and has multi-drug resistant aspects to it. So we looked at the susceptibility to those two groups of organisms and were able to make publications. The next study I did then was to extend the study to look at MRSA and to compare antibiotic resistant organisms to the levels of susceptibility that I found in those first two groups of organisms. And it immediately became clear that if you had an antibiotic sensitive strain of Staph aureus and an MRSA, and I think we had 18 strains of MRSA, they all had the same susceptibility to Manuka honey. So antibiotic resistant organisms, antibiotic sensitive organisms had the same susceptibility to the Manuka honey, which was quite exciting. And a whole range of antibiotic resistant bacteria have now been published, and a nice review that you might be interested in, written by Dee Carter and colleagues from Sydney, was published in 2016, and at that time she was able to list 60 susceptible bacterial strains when it came to Manuka honey. So Manuka honey is no longer an alternative. My first PhD student was tasked with finding out how does Manuka honey inhibit the bacteria, because if you can explain how it works, you've got a better chance of persuading clinicians that maybe they would like to use it. So Anna Henriques did some electron microscopy. I'm a great believer in seeing is believing. So first of all, if you look at this slide, we'll work from the left-hand side to the right-hand side. And the left-hand side, we've got some Staph aureus, as seen by scanning electron microscopy. They've not been treated with honey. The middle image shows that the bacteria have been treated with honey. They're less numerous, but really they don't look a great deal different to the ones on the left. So the surface hasn't much changed, but when you look inside, use transmission electron microscopy, we were really struck with a number of cells that had got a cross wall. Now normally staph will take about 30 minutes to complete the cell cycle, and it's only at the end of the cell cycle you see the cross wall being formed before the bacteria divide. She also looked at Pseudomonas aeruginosa, and again looking at the left hand slide, that's a control not treated with honey. The middle slide has been treated with honey, and immediately you can see a difference in structure. The cells are smaller and they've got irregular surfaces. And then when you look inside the cells with transmission electron microscopy, I think you can agree that again they're misshapen and bits of the cell wall are missing, and there is even lots of there is some debris there. So Anna's study showed that the Manuka honey has different effects on different bacteria, which again is quite unusual for an antimicrobial agent. Moving on to my second PhD student that worked in this area, which was Rowena Jenkins. She moved on to looking at MRSA, and we were fortunate to secure funding from the British Society for Antimicrobial Chemotherapy to investigate the effect of Manuka honey on the cell cycle of MRSA, because we thought that something was happening to the cell cycle. Rowena used electron microscopy, but this time she used a whole range of concentrations and a range of time points over 24 hours. That experiment was a very extensive one. It took about 5 months to complete. She then looked at the effect of Manuka honey on the enzymes that degrade the cell wall and allow the cell to separate. And finally, we looked at the genes that code for those enzymes. And this is just a kind of a page in the book, you know, that we have thousands of electron micrograph images. And if I start at the top left, A is control, no treatment. B is the cells that have been, MRSA, that have been treated with artificial honey. So to prove to people that it's not the sugars in honey that's causing these effects, we made up a honey with the right amount of sugar that you would find in a honey. C, moving to the right-hand side of the top row, is 2.5% Manuka honey. We know that that is not inhibitory, but then if we look at the lower row, D is 5% Manuka, E is 10%, and F is 20%. So these are lethal concentrations of sugars, and if you just compare the top row with the bottom row, you see those cells have got those cross walls that are inhibited, whereas the ones that are not inhibited are carrying on growing and dividing. And also you'll notice that the cells on the bottom row, the ones that are inhibited, are a good deal larger. Now, to cut a long story short, basically we were able to show that these effects arose within the 30 minutes. When we looked at the enzymes that digest the cell wall, those are called autolysins, basically, although the genes were being expressed and the enzyme was being transcribed, actually the enzyme was not active. So basically the enzyme couldn't degrade the cell wall that released the daughter cells. So essentially, Manuka honey in MRSA interrupts cell division. If the cells can't grow and divide, they can't go on to form an infection. The third PhD in this student in this area was Alid Roberts, and he was tasked with working out what does Manuka honey do to Pseudomonas aeruginosa. Now, in these images, we've used a dye called Live Dead, and it's two dyes. The green one stains living cells, and the red one only gets into dead cells. And so the image on the left is a control, untreated cells, lots of green cells, or those one or two red cells. And on the right-hand side of the slide, you see a low power and a high power image of cells, Pseudomonas aeruginosa, that has been treated with Manuka honey. And you can see that the cells, some of them are red, and you can see the contents are leaching out. Basically, that's the nucleic acid coming out the cells, the contents coming out. So essentially the cell wall has been weakened, and the reason for that is the Manuka honey switches off a gene for a protein that's really important in stabilizing the cell wall. So without that stabilizing protein, the cell wall becomes leaky, and in an osmotic environment, which you're going to find with honey, the cells rupture. So you've got osmotic lysis of those cells. So basically, there are two mechanisms in these, quite distinct mechanisms in these two groups of cells. And we confirm that with atomic force microscopy. Cells on the left have not been treated with honey, the ones on the right have, and you can see the blebs forming quite clearly there. Now, the other thing we did is we looked at the effect of Manuka honey on the gene function, and this is a very complicated story, but essentially it does show that not only does the honey kill the organisms, but it reduces the virulence. And one of the genes that was the most affected was a gene that codes for an enterotoxin, downregulated 109 times. Now, that is a very important enzyme, super antigen, and can initiate allergic reactions in human tissue. Similar sort of effect with Pseudomonas aeruginosa. Motility is a marker of virulence, and in the left-hand pictures you see a series of plates with different concentrations of honey. So moving from top left to the bottom, you have no honey, and the bottom picture where you see no movement at all, I think that's got 12% honey. So as you increase the concentration of honey, the bacteria fail to move. The middle images show the untreated cells have got flagella, the treated ones have got less flagella, and again, the actual genes that code for the proteins in the flagella are downregulated. So we've got detailed knowledge of what Manuka honey does to these bacteria. Now, up until now, I've only talked about planktonic bacteria, free-living bacteria. Of course, there's a great deal of interest in biofilms because they're associated with chronic wound chronicity now. So we need to know, does Manuka honey have an effect on biofilms? Several studies had been published before we got involved in this, and the last 3 studies on that slide were actually done in my own laboratory on Pseudomonas and Streptococcus pyogenes and Staph aureus and MRSA and VRE. When bacteria are integrated into a biofilm, they become much less susceptible to all kinds of antimicrobial agents. They become tolerant to those agents. So we can, with relatively low concentrations of Manuka honey, prevent a biofilm forming, but once it's formed, we actually need much higher concentrations. But in the next series of slides, you see we can disrupt a biofilm. Now what we have here is we've grown Pseudomonas aeruginosa on cover slips, and then we've added honey, incubated for 24 hours, and then looked at them. First of all, this is with a scanning electron microscope. So if we start in the top left-hand corner, I'm going to go clockwise around the slide and end up in the middle. So the top left is untreated, and then next to that on the right top tier you've got 20% honey. Bottom tier on the left is 30%, bottom on the right is 40% honey, and in the middle you've got 50% honey. So as you increase the concentration of honey, you can see you increasingly disrupt the biofilm, and certainly if we concentrate on the middle picture, it looks as though that biofilm has been extensively disrupted. But the thing about looking at electron micrographs is you don't still know if the bacteria are alive or dead. So if we do this, and the slides are in the same order here, with the live dead cell, you see at top left an untreated biofilm, lots of living cells, 20% honey next to that on the right top tier. We've already disrupted those clusters. And then as we move up to 30, 40 and 50% honey, you see how the numbers of living organisms diminish quite considerably. Even with 50% honey, there are still 1 or 2 living organisms available there, and these are what we call persister cells. But at least the number has been significantly reduced, and that gives the immune system in the human a better chance of coming along and engulfing those bacteria. And this kind of work has been confirmed by other workers. It's not just what's happened in my laboratory, and here are some other studies. Many of them came from Australia that confirm those kinds of observations. And what's also been done, which is quite nice, is in one study a multi-species biofilm has been used, because we only use pure cultures. It's one thing looking at what happens in the laboratory, but of course we want to know, is this really translatable to the clinical practice? And so here are 2 case studies that were published, and the first one is with a lesion that in fact had MRSA. It was colonized by MRSA. That lady had that lesion for 5 months. Within 2 weeks we cleared the MRSA, and she healed within the 3rd week. And again, the lady on the right hand side again was colonized with MRSA, and we were able to show treatment with topical honey, Manuka honey, that the MRSA was eradicated. And following those observations, others have actually used honey in the same sort of way. So there's evidence that it can be used palliatively in hospice patients with facial wounds. Plastic surgeons seem to be quite happy with it. The 3rd study on that slide was with neonates in Germany, where 7 consecutive patients with oncological conditions were eradicated MRSA. And certainly in venous leg ulcers, Georgina Gethin in Ireland was able to show MRSA being eradicated following treatment with honey. And the last observation on that slide is a recent trial that was published earlier this year, where Medihoney was used versus mupirocin for the nasal decontamination of MRSA, and there was no statistical difference between the 2. So to me that means that they're as good as one another. The problem with mupirocin is if when you use mupirocin, you are likely to select for mupirocin resistance. But we've done some tests with bacteria, and when you use Manuka honey, you do not select for resistance, no known resistance to Manuka honey. And this, again, just to finish off the antibacterial attributes, this was a young lad that, again, was treated before we had licensed products. There was all prototypes at this time. And this boy had suffered from meningococcal septicemia. And the picture you see there is him after 9 months in hospital. Estimated cost about £1 million. All conventional therapies had failed to remove to heal those lesions. There were 3 organisms involved. Staph aureus, I think, was one, Enterococci and Pseudomonas aeruginosa. And the most important thing for that 15-year-old boy was the smell. He was very conscious of the smell and the pain. And so reluctantly, it was a kind of worst-case scenario. What else can we do? He was treated with honey and healed completely within 10 weeks. So that was a phenomenal result. As far as comparing honey with nanocrystalline for control of odour, a study that I think was conducted in Norway has shown no significant difference, but certainly palliative treatment of carcinomas by Drain and Fleming did show improvement in controlling odours. So you see there that Manuka honey has multiple effects. What I'm going to do very quickly now is turn my attention to its possible impact on the healing process. Can it contribute to healing? And first of all, we'll look at debridement. Now, if we start at the left-hand side of the slide, what I've shown you there is a healing wound, and in a healing wound, you've got autolytic debridement. Now, when you get exudation, it brings an inactive form of plasminogen into the wound, which gets activated to plasmin, which is then able to break down fibrin clots and remove any extraneous tissue material on the surface of the wound. Now, in a chronic wound or a stalled wound, that process is interrupted because of the presence of macrophage, which release an inhibitor that prevents the activation of that plasminogen. So basically, you don't get breakdown of fibrin, and you get a lot of non-viable tissue accumulating in the wound. Now, Peter Molen was able to show that if you add honey, topical honey, to a wound, that prevents the release of that inhibitor from the macrophage. So basically, it is able to restore autolytic debridement. It looks a very complicated slide, but I hope you follow my reasoning there. Let's quickly turn to anti-inflammatory effects. And again, this is one of the experiments that Anna Henriques did during her PhD. And that is to quench free radicals. Free radicals are very high levels in chronic wounds, and they can be very damaging. To an extent, all honeys actually, or all sugars even, can mop up radicals. And in the left-hand image there, we have a whole range of sugars and honeys that can mop up the free radicals. And in fact, of the ones that we tested, Manuka honey was the most effective. The second experiment that Anna did in this series here is, if you look at the right-hand panel, she made a solution of Manuka honey, solution of it, and there are no free radicals there. Now, if that was a peroxide generating honey, it would be producing hydrogen peroxide, and there would be spikes in the top trace. So Manuka honey doesn't produce those free radicals. In the middle trace of the the right-hand image, Anna introduced hydrogen peroxide. So immediately she squirted it in. Within seconds, you see the trace, and 5 minutes later, the bottom trace on that right-hand panel, you see that the free radicals have been quenched. Now, quite a lot of work has been done on anti-inflammatory effects of honey. We did one such study in Cardiff, where we were able to show that the production of free radicals was significantly reduced in the presence of Manuka honey. Many people have looked at the components that might be responsible for those anti-inflammatory effects, and quite a few of those studies have been done in New Zealand, looking at the antioxidants, the anti-inflammatory agents, those phytochemicals that I mentioned earlier on seem to be responsible. But the last study in that slide shows some work that was done by Alvarez Suarez, who in fact has got a mechanism, has actually showed a pathway that's involved in those anti-inflammatory effects. And this is a study that was conducted in Cardiff. It was a collaboration between my own university, King Saud University, Saudi Arabia, where Professor Abdullah Al-Angari came to do some sabbatical work with us and brought the funding with him. And the third collaborator was the University of Southampton. And so what we've got here are a mixture of laboratory and in vivo effects. The first was a kind of proof of concept study where we recruited 16 patients that had bilateral atopic dermatitis. Sadly, we lost 2 during the study, so 14 completed. These patients had to have bilateral lesions, so they acted as their own control. And the volunteers were asked to leave one site untreated, and if they were in discomfort, they could resort to their normal treatment. And the other site, and usually it was the one with the worst symptoms, clinical symptoms, was treated overnight for 7 consecutive nights with Medihoney and covered with gauze. Then in the morning all they had to do was wash it off. The Professor Al-Angari evaluated the clinical symptoms using a TIS score, and we were able to see that if you look at the slide, the graph, the first 2 columns on the left are controls before and then controls after the 7 days. So these patients didn't receive the honey, and there was no improvement, no change in their clinical symptoms. The 2 lines on the right-hand side of that graph, the third one is the patients before treatment, and then the last one on the right are the clinical symptoms judged after the 7 days. And the statistically significant reduction in clinical symptoms was found. Just to show you one example, what we have here is a patient at the top at day 0. So the left-hand top image is the untreated, the lesion we decided not to treat, and you see the one on the right at the top is the one that was slightly worse, and so we decided to treat that. And so in the bottom of this panel you see the results after 7 days, and the image on the left, there's no improvement in clinical symptoms, but there is in fact in the right. And in fact this patient told us the night before she came to be reassessed on day 7, she realized that instead of watching the television and scratching both knees, she was only scratching one knee. So you can see that there is an improvement in the symptoms there. Certainly that last picture on the right looks a lot less angry. So we also did some laboratory work, and I'll just summarize one aspect of this, and where in fact using mast cells and Medihoney, it was shown that the release of histamine was significantly reduced. So degranulation of mast cells was significantly improved using the Medihoney. Pro-inflammatory activity has also been associated with Manuka honey. And I should remind you that you might think that that perhaps is not a good thing, but the natural healing process is a whole sequence of events, pro-inflammatory and anti-inflammatory activity, that has to work in balance. And so we have been able to show that Manuka honey does stimulate the release of TNF-alpha and IL-1 beta and IL-6 from monocytes. And similar observations of stimulation of cytokine release has been demonstrated with the French honey. Now, I've covered an awful lot of time in a very short amount of time. I've basically told you about the antibacterial components and the wound healing potential of honey. But you don't any longer have to believe only myself. And I was really happy to find this on the website of NICE, the National Institute for Health and Clinical Excellence, which is a very august body in the UK. And basically what they're saying here is that medical grade honey has antimicrobial and anti-inflammatory properties and can be used for acute and chronic wounds. So to finish my part of the presentation, I've shown you the antiseptic properties, talked about the broad range of antimicrobial activity, but concentrated on mostly the work that we've done in my own laboratory with gram-positives and gram-negatives. Shown you that honey has different effects on different bacteria, that it can inhibit biofilms in vitro, but in vivo I've shown you that it can eradicate MRSA from colonized wounds. Now with infected wounds, of course, you do need antibiotics. You couldn't just rely on Manuka honey there. And that Manuka honey has some evidence of controlling malodor. With the wound healing effects, its osmotic potential creates a moist environment because it draws fluid out of the wound. That in itself helps to reduce any edema which might be associated with pain. It does have debriding action, anti-inflammatory action, and in addition to that, it can stimulate those cells involved in the wound healing. And there's a very nice review written some time ago by Uri Matjan talking about the immunomodulatory properties of honey. So with that, I'm going to hand you over to Lisa, who's going to talk to you about some clinical effects.
Hello, my name is Lisa Sangster and I'm a clinical nurse educator in northern Nova Scotia. I cover a population of approximately 40,000 people. I don't have a strong background in education. I haven't taken any credentialed courses. However, I love wound care, and so I've taken it upon myself to learn as much as I can about the wound care products that we have available and how to treat various wounds. As the clinical nurse educator, I use this knowledge to assist others in determining how to best treat wounds. Today I'm going to share with you some experiences that we've had with Medihoney. So my story begins with Cooper. So Cooper is my dog here. And approximately 4 years ago, Cooper was bitten by another dog while I was away. And when I came home approximately 14 hours after he'd been bitten, I discovered that he was quite lethargic. He had 2 puncture wounds to his neck. It was red, it was swollen, and he had some purulent drainage to it. So I had to really think about what did I want to treat my 18-pound dog with. I knew that I wanted to choose something that was non-toxic, because, you know, I didn't want to make the situation worse. So I decided that Medihoney would be where I would go because of its natural properties and it being non-toxic. Also, I chose actually a Medihoney wound gel because the wound gel contains a gelling agent that would help me to keep the product where I wanted it and not basically go everywhere. And most importantly, it was really easy to apply because you can imagine it's a nightmare to try to get your, you know, he wasn't going to let me shave him to get this wound care product on him. And I wanted something that was going to stay at the wound bed and I wouldn't need to have an adhesive dressing over top of it. I did, however, apply a cling wrap to his neck very loosely. Don't worry about that. Mostly just because I didn't want him to rub the honey all over my furniture while we slept. So after about 8 hours, all the signs of inflammation were gone. There was almost no drainage at that time. He had regained a lot of his energy, and I was very pleased with that, and it just really sold me on the product. However, I decided that I didn't want to take any chances, so I continued with the use of the Medihoney gel for 4 days, washing it off each day and reapplying it. And after 4 days, he was completely healthy, and I'm happy to say he's 16 years old and still with us. So why did I choose Medihoney, and why do I choose Medihoney now in my practice? Well, I choose it because it's so easy to use. That is probably the biggest reason why I use Medihoney. I don't have to worry about it harming anyone. It's very non-toxic, it's natural, and it's easy to use. As Dr. Cooper has mentioned a lot of these things previously, but it is effective against a broad spectrum of wound pathogens, including antibiotic-resistant strains of MRSA and VRE. You can imagine with my dog Cooper, I had no idea what kind of organisms were growing there. So again, I knew that the honey would treat him. It's effective against biofilms. It reduces the risk of infection. It's anti-inflammatory. It reduces edema and provides a moist wound environment. It debrides the wounds, and it helps lower the pH of the wound. In my first human case study I'm going to talk about, I'm going to share with you this gentleman. He's a 47-year-old diabetic male. He presented for his yearly appointment at the diabetic clinic to be evaluated, and upon examination we found this deep fissure in his top of his right great toe, and both of his feet were extremely dry. According to the patient, the crack had been there for approximately 8 months, but he wasn't overly concerned about it because, to quote him, it was just dry skin and it wasn't causing him any pain. For treatment, the physician decided to mechanically debride the toe, removing the majority of the callus. Another option would have been that we could have applied the Medihoney, and it would have eventually debrided away at that wound, but by debriding it we were able to speed up the healing process. After the debridement, we decided that we would cover the entire feet with the Medihoney barrier cream as a protection, and it would also help to moisturize the foot because of the osmotic effect it would cause, bringing moisture to the wound surface. And for the toe itself, where the fissure was created, we applied a Medihoney wound gel. Again, we stuck with the Medihoney wound gel because it was simple to apply, and it's a little bit thicker than the 100% honey, so it stays right on the site. Because there was very little drainage with this wound, we chose to just have the gentleman wear his socks daily, and we weren't concerned about it. So he came back to our clinic every 3 days for 3 weeks, and you will see from the photo on the very bottom there that the wound has completely healed. And we have encouraged him to continue to use the Medihoney barrier cream as a preventative to keep from this happening in the future. This next case study concerns a 53-year-old plumber who received a chemical burn to his right foot and his lower inner leg. The patient did not seek medical treatment for 5 days. He admitted that during those 5 days he didn't do anything to treat the burns himself. In fact, he had hidden it from his wife. His medical history was insignificant, and he had no history of diabetes, venous or arterial disease. When he presented for treatment, there were several areas of necrotic tissue and some slough noted along the edges. After cleaning the wound, we decided to apply a Medihoney barrier cream to the entire foot and lower leg to maintain the skin's barrier properties, and to also lower the skin pH in an attempt to reduce the inflammation and potential pathogen growth. We then covered the wounds with a thick coat of 100% honey liquid. In this form, the Medihoney liquid would quickly penetrate the necrotic tissue, and it would act faster. The Medihoney, with its acidic pH, would also draw the fluid out more quickly so that the necrotic tissue would eventually debride. We covered the Medihoney with Medihoney aponate, again making sure that there was a lot of Medihoney still on the wound. So why did we do this? Well, the reason I like to do both the Medihoney 100% and the aponate is the fact that I want it to start acting very quickly. So the 100% Medihoney starts acting very quickly because it's already in absorbable form, whereas the aponate would take a while for the Medihoney to be released from that dressing. And we instructed this gentleman to return every 3 days. So you'll see here from these slides that on day 4, the areas were starting to debride. There was a copious amount of drainage, but because we had put the extra-zorb dressing on it, there was no skin maceration. And so we continued with the treatment of Medihoney barrier cream the 100% Medihoney, and the Medihoney Apinate, and again we covered it with the Extra Zorb and held it in place with Cling. Again, we instructed him to come back in 3 days. So here's a photo of that same burn. Now it's day 12. You'll see that the dead tissue is fully debrided and new granulation tissue has started. The treatment we decided was to continue with the exact same dressing, but we did go to a lesser absorbency Exorb simply because there was a lot less drainage, and the lesser absorbent dressing was a little bit more pliable on the foot, so it didn't seem quite as bulky for the patient. And then we have the chemical burn on day 24. The areas continue to heal well, and we continue with the exact same treatment because why change what works? And then we have a photo at the top here of the chemical burn on day 31 and again on day 38. You'll see on day 38 that it is completely healed. But we did encourage the patient to continue to use the barrier cream daily because it also has the properties of reducing scarring. And we discharged him from our service. This next case study involved a 53-year-old woman who presented with this wound to her right ankle. She had no idea how she initially gotten the wound, and we kind of assumed initially that it was vascular, so we checked her ABIs and found that they were normal. She was not a diabetic and she had no significant medical history. The ankle was red, swollen, and warm to touch, and as you can see there were some small varicosities on the lower edge of her foot that we attributed to her occupation. On a pain scale, she actually said this was a 5 out of 10, and she had told us that she had tried polysporin at home with no effect. So when she presented to us, we started her on Keflix for 1 week, and we cleansed the wound, and we applied, of course, my favorite, a Medihoney barrier cream to the entire foot. Then we applied a thick coat of Medihoney 100% directly on the wound. We covered that with Medihoney Apinate and an extra dressing and cling wrap to hold in place. All of this was chosen because we were trying to provide a moist wound environment, encourage granulation tissue formation, and we wanted to remove the dead tissue and exudate from the wound. So the following slide shows day 3, day 8, and day 14. They show a gradual progression of the wound healing. Each one shows a decrease in redness, inflammation, and the wound exudate, and an increase in granulation tissue. I particularly like this slide from picture from day 19 here, because you can see the granulation tissue really starting to form, and you can see that the peri-wound skin it's starting to, the soldiers are marching from the edge into the inside, and there's no peri-wound damage being caused by the exudate. Our most recent photo is actually just recently, and that's day 22 on the far right-hand side, and you'll see that there is a great reduction in the amount of the size of the wound. So we have continued to treat her with the barrier cream, the Medihoney 100%, and the Medihoney Apinate, the extra shrub dressing, and a cling wrap. And we're quite pleased with those results. So those were just a few examples I had to share. I'm also going to share with you this algorithm that was provided to me by Dermascience. I'm not going to go into great details about it, but it clearly tells you that you have to determine what your type of wound is and what your objective is so that you know what dressing is the proper one to use. So this is just a little algorithm that I have in my toolbox that I pull aside when I need it. As such, I've used Medihoney on different types of wounds, including those not included in the presentation. I have not seen any evidence to support the use of Medihoney in third-degree burns or in conjunction with other antimicrobial products. However, I believe there is research being done on that right now. And of course, I wouldn't use honey on any clients that had an allergy to the dressing material or the honey. And this is a question I'm actually I get a lot, is I'm allergic to bees. Can I still have the Medihoney? And yes, you can. So as you can see, there's a few products that were listed on the algorithm. I'll go back again. and they were the Medihoney gel sheet, the Medihoney tool, and the Medihoney HCS. And I haven't talked about those. I have used all of these products. I just happen not to have a case study for them. So if you'll just give me a moment, I'll tell you a little bit about the Medihoney gel sheet. The gel sheet is pretty much exactly like the gel, only it comes more in a dressing. It comes in a sheet. And the only reason why I don't use the gel sheet all the time is the fact that I want the honey to actually touch the wound bed, because in order for the honey to be effective, it has to cover the wound bed. The gel sheet is flat. It has so it's going to lay over the top of the wound. So when I do use the gel sheet, I tend to fill the wound with Medihoney gel or 100% Medihoney and put the gel sheet on top. By doing so, I've made contact with all of the wound bed, and I've also added the extra honey that the wound will need if it is a highly exudating wound and I want the honey to last longer. The benefit also of the gel sheet is that the gel sheet goes to work as it reaches the body's temperature, and if you will, for better words, as it melts away or the honey is released from the gel sheet, that's when it would come into work. So by adding the honey first, we are actually causing the product to start to work faster, and by using both, we're allowing the honey to stay on the wound bed and last longer. The other product that I have used before is the Medihoney tool dressing. The thing with the Medihoney tool dressing is a non-adherent dressing, and when I have used this has been on very delicate wounds when I wanted to put something on the wound bed to protect it. The great thing about the Medihoney tool sheet is that one, is impregnated with the Medihoney, but it can be left in place for 7 days. So when I've used this in the past, if I had to change the secondary dressing because of increased exudate, I would actually remove the top dressing, add more honey to it, and then put another secondary dressing on top. And again, I would just change that at 7-day mark. The last product I'm going to mention is the Medihoney hydrogel colloid sheet. So what this is, it's an all-in-one dressing that contains the Medihoney in a hydrocolloid hydrogel sheet, and it can actually handle 2.5 times more fluid compared to other hydrocolloid dressings. This is really good use for moderate to dry exudating wounds. And it's lovely because it comes in 3 different formats. You can get adhesive, non-adhesive, and fenestrated. It has very non-traumatic removal, and it delivers a cooling, soothing effect when you first put it on the patient. Again, this is something that does take a little bit of time to release the product as it gets to that proper room temperature. So when you're using any of these Medihoney products, you have to be cognizant of the fact of how often you're changing your wound dressings, because if you think about thermoregulation, you don't want the wound to become cold and then have to get back to that body temperature again. So if the product says keep it on place for 3 days, keep it on place for 3 days. That's what it's intended for. So how does Medihoney work to eliminate wound odors? Well, Dr. Cooper mentioned this a little bit, but basically the bacteria in the wound is consumed by proteins, consumes proteins of the tissues and the serum, which gives you off the odor or byproducts. And when the Medihoney is produced, the bacteria begins to consume the glucose instead, thus eliminating the odor, and there's no more byproducts. It also inhibits or eliminates bacteria, which again eliminates the source of odor. Now, this is one that I really had a lot of interest in, and that was for the use of chronic wounds. If you think back to your chemistry days, pH is the measure of hydrogen ions concentrations in a solution. So solutions with high concentrations of ions have low pHs, and solutions with low concentrations of hydrogen ions have high pH. Some examples, and I always find this so interesting, is Coke and orange juice has a pH of about 2. Water is around 7. Baking soda is around 9. And household bleach is around 12. So, ironically, your skin fits kind of more on the acidic side. It's about 5.5. And that helps, when your skin is at that level, it helps keeps the lipids and moisture in while blocking germs, pollutions, toxics, and bacteria. So interesting enough, if you change the pH of your skin by, say, maybe some skincare products that you're using, or in this case, we're talking about the Medihoney Barrier Cream, by adding it to the skin, we are lowering the pH so that we're actually improving the likelihood that we're going to keep the lipids and moisture in and block those germs and pollutions and toxins. When your skin is too alkaline, the skin becomes dry, sensitive, and you may experience inflammation, which inhibits the skin's ability to ward off pathogens, and chronic wounds usually have a pH that is high. So when we use Medihoney, which has a pH of, Dr. Cooper said 3.9, I'm going to say 3.5 to 4.5 range, we alter the pH level of the wound by creating an osmotic effect that causes fluid to move from the deeper tissues to the wound surface, which promotes the wound healing. So I have found that Medihoney is very effective in those chronic wounds that I have not been able to do anything else with. So I do have a few tips that I'd like to share with you now to end my presentation. So please use only Medihoney that is especially medical grade Medihoney and not your household honey. Ironically, and I never had a chance to speak to anyone about this before, but one of the other reasons why I really like the use of honey is the fact that some of our clinicians in our area order icing sugar to be put on the wound. And we have had a lot of success with it, but it was very interesting to look at the properties that Dr. Cooper outlined about the sterility, for instance, of the product. So that is one of the reasons why I wanted to introduce Medihoney to our organization, because the nurses were often concerned about how effective of a medical product was actually going to the kitchen and getting icing sugar, and how do you prepare it. The other thing I've alluded to is that you need to ensure that the honey is in full contact with the wound bed, and so you need to fill any cavities and any depressions. So again, if you're using the gel, put something underneath it. And if you're going to put anything into the tunnels, it's best to use 100% liquid honey rather than any gel products or anything else, because it could get stuck in there and is difficult to get out. If using a non-adherent dressing between the honey and the wound bed, consider using Medihoney Tool. And if you're not going to use Medihoney Tool, if it's something that you don't have available and you have another non-adherent dressing, just make sure that it's porous enough to allow the active components of the honey to diffuse through it. Please ensure that dressings extend to cover any area of inflammation surrounding the wound. We are not overly concerned about it causing any problems because, again, it's going to create that moist wound environment, and it's going to help reduce any kinds of infections. So it's not a concern about healthy tissue. Use a suitable secondary dressing to hold the honey in place and to prevent leakage of the honey and prevent skin maceration. And pressure bandages can be used over the honey dressing. Always remember to change the honey dressing frequently enough to prevent the honey from being washed away or diluted. And if because it could stick to the skin if there has been left too long. So be sure that you can always add a little bit of normal saline to it if you need to moisten it. Applying 100% Medihoney directly on the wound bed in addition to another honey dressing will increase the initiation of the therapy. We talked about that. And lastly, I think the most important thing is that you need to educate your patients about why Medihoney works. To them, it seems quite strange to be actually adding honey. They think that you're adding something from the grocery store, and it's very important that you let them know that it's not from the grocery store, because you don't want them to do that the next time. And when I'm doing my education with the clients, I also talk about a stinging sensation. I have had a few clients complain that there was a little bit of stinging sensation when it was applied. It usually only lasts about 30 minutes, and usually they can tolerate this if you've explained to them in advance that might happen. And that's just simply because the honey is doing its work. It's starting that osmotic effect and drawing the fluids to the tissues. But it can be minimized by preparing the patient and pre-medicating, especially on wounds that are particularly sensitive. And in my experience, if you use something like the Medihoney gel sheet, it works well in preventing the pain because there's more gradual absorption of the Medihoney as it's released by the body's temperature. So to summarize, I'd just like to say, to quote a commercial that you all might remember, I put that on everything.
Thank you both very much. We've heard from Professor Cooper about the science of Manuka honey and its impact on wound healing from Lisa Sangster on her clinical perspective and use of Medihoney here in Canada. I wonder if we have any questions from the viewing audience.
Thank you to the speakers for the information. We are now going to move on to taking some live questions from the audience. Our very first question is Manuka honey a real and effective alternative to silver?
Well, maybe I can answer that. Of course, both silver and Manuka honey are antibacterial agents. To my mind, one of the advantages of Manuka honey is that it's never ever been shown to be cytotoxic, and certainly there are publications in that field. And the other important thing, I alluded to it briefly in my presentation, we have done some training experiments in the laboratory to try and select for honey resistance, and that we've done that by exposing bacteria to low levels of honey over a 28-day period, and we have not been able to isolate honey-resistant strains. Similar work has been done by Shona Blair in Sydney and Paulus Kwakman in the Netherlands, also having looked at a range of bacteria, has come to the conclusion that there is no resistance to honey. So I think those are two important points.
Thank you. Any other questions?
Yes, we do have. Our next question is, there are so many different products and they all boast at being the best. How do you decide what product to use?
I'll take this one. You're right. There are a lot of products on the market, and they all think and they all talk about being the best. And choosing the dressing is always a difficult decision. When presented with a wound that people want my help in determining how to treat it, I have certain questions that they have to answer. What is the cause of the wound? Where is it located? How big and how deep is it? Is there any tunneling? What does the wound bed look like? Is it necrotic? Is it good granulation tissue already? Is it dry? Is it wet? How much drainage is there? What does the peri-wound skin look like? Is there inflammation? And is there an odor or is it painful? And most importantly, all of my nurses know now that I want to know about the patient, because I want to know what are our expectations? What are we trying to accomplish here? Are we going to be able to heal this wound? If you answer any of those, all of those questions, I should say, you can greatly narrow down your choices by determining what you want to do and what are the attributes of the wounds, attributes that the wounds have that you want to take care of. But it's still a guessing game. I use a lot of products, and I think what happens is that you go back to your memory to the time where you've used a product successfully, or you've had a similar situation, another client with the similar wounds, and you think about what worked well at that time and what didn't work well, and you make the determination based on that. So that helps to narrow it down, but it's still a guessing game. The big thing I'd say is don't flip from product to product. Choose a product and give whatever product you chose a chance to work. Know that with any product, and I know Medihoney will even say this in their studies, is that sometimes it does look worse before it looks better. If you think about what your wound bed looks like and you're using the Medihoney effectively, you're going to be drawing out some of those tissues, and it may look a little bit bigger and it may actually look worse. So give it a chance to work. And don't flip from product to product. And I always recommend that you need to stick with a product for at least 2 to 3 weeks to actually give it a chance to work. And also, follow the directions for the product. Make sure you know what that product is so you've chosen the right product, but you use it according to how they've told you to use it. So if they've told you that the product can be left on for 3 days, and we're talking like, say, the Medihoney gel sheets now, leave it on for 3 days, because taking it off earlier than that, you're going to have that thermal problem again with the wound getting warm and cold. So you're not using the product appropriately, so obviously it's not going to work. So follow the directions. And of course, a lot of time, it is just, our choice is affected by what is on the shelf. Many of us work in large organizations that the stock is provided to us, and we have to use what's available to us. But fortunately, I have not had any issue getting Medihoney because it is very cost effective, and it does not have to be, it's not a dressing that has to be changed daily, so nursing staff time is greatly reduced, and the extrazone dressings are great because they wick away and keep the wound from getting macerated.
Thank you. Additional questions?
Our last question is, how can clinicians be sure of how much methyl is in each Medihoney dressing?
Well, with the methyl certainly the honey will be tested at the hive. It'll be tested throughout the production process. And I understand also that once the products have been produced, they'll be retested. So you can be assured that the level of methyl that's claimed, that's the 355 milligrams per kilo of honey, has been verified, and it is there. If I could add something to the first question that I answered, I'm sitting here and thinking I could have said perhaps a little more. And one of the things I would say about one of the advantages of honey compared to other antimicrobial agents is, of course, that honey not only is an antibacterial agent, but it also has an impact, a positive impact, on wound healing characteristics.
Wonderful. Are there any? There are some more?
Our next question is, if the stinging lasts for more than 30 minutes, then should you completely stop using the product?
No, I wouldn't stop using the product. I would probably try to add a little bit, maybe go with the gel that has the gelling agent in it versus the 100% honey. Now the gelling agent is still 100% honey, but there's 80% of that is honey and 20% is the gelling agent. So a lot of the stinging will go away if you switch to a lesser product. I don't mean lesser, not quality product, just lesser concentration of product.
I hope we have some other questions.
Our next question is, between silver and Medihoney, which is most cost effective?
Well, I actually don't look a lot at the pricing of these things, but I do know that Medihoney is very reasonably priced, and I think it depends on your contract and where you're purchasing it. For me, Medihoney is a better price, and so I have never had any issue getting it.
Actually, I have a question. I believe it was the FDA that suggested that the unique Manuka factor should be between 12 and 16, and we know the Medihoney products are above 12. Mm-hmm. Are there products that are higher rated than that in terms of, does it come with a higher Manuka factor, unique Manuka factor?
If you were buying honey to consume, certainly you could buy UMF above that, but that would be in jars of honey for consumption. I don't know of any medical products that contain higher levels of MGO. Mm-hmm. And certainly if you go to other company products that don't have the Manuka honey, they won't have MGO in them anyway.
Mm-hmm. Great, thank you.
Our next question is, Can you use Medihoney on diabetic foot ulcers in patients with high glycemic values?
Yes. Plain and simple, yes. It's going to be broken down, and it's really not going to affect the diabetic.
I actually wrote a review that was published in the Diabetic Foot Journal in September 2017, and I did cite 3 studies there that showed that honey was safe to use and didn't affect glycemic levels in diabetic patients. I can't actually remember the citations now.
We do not have any more questions at this moment. Thank you very much to the speakers for answering the questions. I'm now going to flip it back to Andrew for concluding remarks.
I want to thank our speakers very much for their very interesting and informative presentations. I could have asked questions myself here for the next half an hour, but we don't have that amount of time. I'm sure that there are other viewers out there that will have further questions. There will be a slide showing details on how to contact an Integra representative, so please feel free to do so and contact your Integra, your local representative, to get more information. Thank you very much for tuning in.